Hello class, today we are talking about ELISA test. ELISA, which stands for Enzyme-Linked Immunosorbent Assay, was first developed in 1971 by two Swedish scientists, Eva Engvall and Peter Perlman at Stockholm University. This technique is used to detect and quantify substances such as peptides, proteins, antibodies, and hormones that are indicated by protein and antigen. Because of this trait, it is used as a diagnostic tool in different kinds of science fields such as medicine, plant pathology, and biotechnology. But before we start, let's go over some key terms. Antibody, also called immunoglobulin, is a protective protein produced by the immune system in response to the presence of a foreign substance called an antigen. How does it work, you may ask? The ELISA test works by the use of antibodies which aid in detecting the presence of a specific antigen in a sample. In an ELISA, the enzyme is attached to the antibody. Antibodies are usually constructed in response to some sort of infection. So the use of this test can assist in determining if the sample has had contact with a specific virus. But what is it used for? Well, ELISA tests can be used in a scientific world for diagnosing real-life diseases, infections, and viruses. For example, the ELISA assay is the most common test in diagnosing HIV, which causes AIDS. Through collecting an individual's blood sample for antibodies as well as oral fluid, through the use of this unique technique, it can also be used for confirming syphilis infections, Lyme disease, and it is also found in home pregnancy tests, just to name a few of the more common purposes of this assay. Throughout this presentation, we are going to be going over the 4 different types of ELISA tests, which include direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So to start off, the first test we will be discussing is direct ELISA. Direct ELISA is the attachment of an antigen to a polystyrene plate followed by an enzyme-labeled antibody that can react with the antigen and substrate that can be measured. Direct ELISA consists of 4 steps: coding, blocking, detection, and reading the results. The first step in direct ELISA is the antigen is immobilized onto the wells of 96-well polystyrene plate via passive absorption. The second step involves an enzyme-labeled primary antibody, example, HRP-labeled primary antibody, specific for the target antigen, is added to the wells and directly binds to the antigen. And in the final step, a respective enzyme substrate is added, which upon reaction with the enzyme produces a visible colorimetric output that can be measured by a spectrophotometer or absorbance microplate reader. The second test we will be discussing is indirect ELISA. Indirect ELISA is the attachment of an antigen to a polystyrene plate, followed by an unlabeled primary antibody, followed by an enzyme-labeled antibody that can react with both primary antibody and substrate. The first step in direct ELISA is coat ELISA plate with testing antigens, seal the plate, and incubate overnight at 4 degrees Celsius. We then remove coating solution and wash plate 2 times with desired puffer. Block plate with desired blocking solution for 1 hour at 4 degrees Celsius and wash plate 2 times with buffer. Incubate with unconjugated primary antibody at room temperature for 1 hour, wash plate with wash buffer. Incubate with HRP labeled secondary antibody at room temperature for 1 hour and wash plate with buffer. Incubate plate with TMB substrate solution at room temperature for 15 to 30 minutes and stop reaction using HRP reaction stopping solution. We then measure the absorbance signal at 650 nanometers with an ELISA microplate reader. The third test we will be discussing is sandwich ELISA. Sandwich ELISA is a capture antibody is attached to the polystyrene plate, then antigen is added that specifically attaches or captures the antigen. A second antibody, also specific for the antigen but not the same as the capture antibody, is added and sandwiches the antigen. This second antibody is then followed by an enzyme labeled antibody-specific for the second antibody that can react with a substrate that can be measured. The first step in this procedure is the plate is coated with a suitable capture antibody. Blocking buffer is then added to block remaining protein binding sites on the plate. The second step, a sample is added to the plate and any antigen present is bound by the capture antibody. A suitable biotin labeled detection antibody is added to the plate and also binds to any antigen present in the well. Avidin, HRP, is added and binds the biotin labeled detection antibody. TMB substrate is added and converted by HRPO to a detectable form. And the final test we will be discussing is competitive ELISA. Similar to sandwich ELISA, but involves the addition of competing antibodies or proteins when the second antibody is added. This results in decrease in the substrate signal generated. This test gives good, highly specific results, so it makes the test the most used out of the ELISAs. In the first step of this procedure, the primary antibody, which is unlabeled, is incubated with the sample antigen. Antibody-antigen complexes are then added to the well plates, which are pre-coated with the same antigen. Unbound antibody is removed by washing plate. The more antigen in sample, less antibody will be able to bind to the antigen in well, hence competition. The secondary antibody that is specific to the primary antibody and conjugated with an enzyme is added. A substrate is added and remaining enzyme elicit a chromogenetic or fluorescent signal. Since the development of the ELISA test, it has become an important and useful tool in a variety of science fields. For a common use, it is found in a hospital. Doctors order ELISA as a screening tool for more in-depth tests because of its convenience. It is a simple test and takes a short time for results, which is why it is a highly useful tool in doctors' offices. Also, the 4 different types of ELISA tests allow the test to be more accurate and versatile in different situations. Therefore, ELISA is a major technique to use in a biological science field. Thank you so much for your time, and I hope you enjoyed our presentation.